perfecto. Voy a presentar al doctor Seb Williams. Seb Williams es una persona muy conocida. Él trabaja en la Universidad de Columbia en New York, en Estados Unidos. Es el Wendy Herons Associate Professor en Women's Health y es el jefe de la División de Reproductiva Endocrinológica en la Universidad de Columbia Irving Medical Center y es el director del Centro de Fertilidad en esa misma institución. Tiene ambos títulos, tiene un título de MD y un título de PhD. El título de PhD lo sacó en Biología Molecular y Bioquímica en la Escuela de Mount Sinai de Medicina. Luego fue al Brigham and Women's Hospital en Massachusetts, que es ahí en Harvard, donde hizo su residencia de ginecología y obstetricia, luego una beca o una fellowship en Reproductive Endocrinology en Weill Cornell, y también otro fellowship en biología de ARN, RNA, con el doctor Thomas Tucci en la Universidad Rockefeller. Él es un doctor científico apoyado, esponsoreado por la NIH, y su focus fundamental es en ayudar a aquellos que sufren de pérdidas frecuentes de embarazo y desarrollar nuevos tratamientos para tratamientos de fertilidad en pacientes infértiles. El doctor Williams yo lo he conocido por una serie de interacciones que hemos tenido entre nosotros y que vamos a seguir teniéndolas. Y él publicó un artículo fundamental en el New England Journal of Medicine, que es el mismo título que la charla que nos va a dar hoy, que va a ser en inglés obviamente, que se llama Rapid Nanopore Sequencing for Aneuplody in Reproductive Care. Ese es el título del artículo en el New England Journal of Medicine, que es aproximadamente hace 9 meses, un año. Un artículo que ha revolucionado todo lo que es diagnóstico embrionario pre-transferencia. El doctor Seb Williams es una persona de altísimo nivel y creo que es un lujo para nuestro colegio tenerlo aquí. What I just said, Zeb, is I introduce you with all your bio, and I said your important contribution in the New England Journal of Medicine recently, about a year ago. And I told all the audience here that it's a luxury for us to have you with us and present all your data that I'm sure is going to be very exciting. So without further saying, I'm introducing you, and I would love you to be here, but I understand the impossibilities, but please go ahead and welcome.
Gracias. Buenos días. Me siento honrado de unirme a usted. Lo siento, no puede estar allí en persona. Ahora tendré que hablar en inglés para el resto. Dr. Ash, thank you for the very kind introduction. It's really a great privilege and pleasure to be with all of you, and especially to be invited by Dr. Ash, who I know in New York and in this country is a very highly regarded and respected professor, and I've heard his name a lot over the years in terms of his contributions, and it's really a delight to be here with all of you, even if it's only via Zoom. What I'd like to speak about in the next few minutes is a new technology that I think has applications across reproductive medicine with the potential to really be able to transform what we do, to expand access, to reduce costs, and to be able to deliver care at a time frame and within a time span that can really change the options of what we can offer to patients. As we all know, one of the central problems across all of reproductive medicine is the issue of aneuploidy, having extra or missing chromosomes. This is a cause of the vast majority of pregnancy losses. It is the primary reason that we do prenatal testing with things like amniocentesis, chorionic villus sampling. It's the reason we check biopsies of embryos to ensure that they have a good likelihood of implanting and resulting in a healthy baby. So testing for aneuploidy, the presence of extra or missing chromosomes, truly spans the entire spectrum from the very earliest embryo through to early pregnancy testing and testing after a pregnancy loss. So if this is such a central problem, such a central issue across all of reproductive medicine, what are the options that currently exist for doing testing? Currently, there's 4 primary options. The first and the most historic is to do what's called G-band karyotyping, where cells are grown, and these cells can derive from a CVS, an amniocentesis, or after a pregnancy loss. The cells are grown, sometimes for weeks, in a specialized laboratory, and then a drug is given to cause the cells to arrest their growth at metaphase when the chromosomes are tightly bundled. Then the cells are stained with Giemsa stain, which is why it's the G in G-band karyotyping. And that gives a little banding pattern to each chromosome, which allows the cytopathologist to recognize which chromosome it is, align the chromosomes, and identify if there's an extra or missing chromosome. Another technology that's commonly used is FISH, where you'll take cells and use fluorescently labeled probes targeting some subgroup of the chromosomes, typically 5 chromosomes. More recently, 2 other technologies have emerged. One is called microarray, where you take DNA and it binds to a glass plate which has little fragments of DNA that are complementary to it to allow it to bind. And it allows you to look at the relative abundance of DNA across all chromosomes. And the last technology that's being used is called next generation sequencing. And I'm showing you a picture of one of the next generation sequencers below. Each of these methods have their strengths and their weaknesses. G-band karyotyping, for example, requires you to have living cells, but it can take weeks to get results back. FISH gives you results back in usually as quick as 24 hours, but it's only looking at a small subset of chromosomes, and as you'll see, it can have a lot of errors to it. Microarray does not require you to have living cells, but it can still take weeks to get results back and is very expensive, often costing in the thousands of dollars. Next generation sequencing, like microarray, can give you results back in about a week to 10 days, but is also very expensive. And all of these technologies have the central problem that they have to be run in a high complexity laboratory. That requires one to take the tissue and ship it to the lab where the testing will be done. Just doing that obviously imparts a degree of delay in terms of being able to give patients their results. It also increases the cost and the risk that samples could get lost or misplaced during transport. So the need to have a centralized high complexity laboratory to run these expensive tests means that in the case of miscarriage samples, at least in this country, I don't know how it is, and in Canada, I don't know how it is in Mexico, But typically, a woman will have to have more than 2 miscarriages before insurance will cover the cost of testing. In the case of embryos, the significance is much greater, because since the biopsy sample has to be sent to a centralized laboratory, and since the testing can take a week to 10 days before results are back, it means the embryo itself must be frozen. And the woman has to both wait an extra month before she can do an embryo transfer and pay the cost to do another frozen embryo transfer cycle. So one can't just take the embryo and put it back in the uterus, because it takes so long to get the results back. And this adds the time burden and expense burden to the patient. In addition, if all the embryos that are tested turn out to be genetically abnormal, meaning they have extra or missing chromosomes, it also means that that embryo, the woman has now paid to have frozen, and now will have to have it just discarded. So she's added, it's almost, the expression we use here is like salt on the wound. She's had something unfortunate happen, which is that all her embryos are abnormal. But on top of that, she's paid often thousands of dollars to freeze those abnormal embryos, only to have to discard them in the future. So what did we do? We turned to look at this very interesting new technology that was emerging in the world of DNA sequencing. And this technology is called nanopore-based DNA sequencing. I'm showing a little cartoon here about how nanopore-based DNA sequencing works. Essentially, you have a little array with these tiny nanopores on it, and that's what you see here in sort of the blue and green. There's a little protein nanopore arranged on a little film, and the strand of DNA travels through that pore. As the DNA is going through the pore, each letter in the DNA, each nucleotide, gives a little difference in voltage. And that voltage generates the little squiggles you see off to the side. Those squiggles can then be interpreted to tell which nucleotide it is. That means that nanopore-based sequencing is actually sequencing in real time. You're seeing each letter, A, G, G, T, C, A, G, T, C, from each pore as the sequencing is happening. Now, why? I mean, that's certainly very cool. And it allows one to sequence very long strands of DNA. So just by way of comparison, the sequencing that you do with next generation sequencing is usually about 200 base pairs long, 200. Nanopore-based sequencing can sequence not 200, but 200,000 nucleotides in length, a single very, very long strand of DNA. And that's what this technology was developed for, to allow one to assemble genomes. For example, the genome of a mouse, a rat, a hamster, food species, and a lot of other animal species. That's what it was developed for, by doing this long-read sequencing. So how do you use sequencing to detect if aneuploidy is present? So how are we doing it today? Next generation sequencing is being used for aneuploidy testing, and I'm going to show you the general concept behind how that's done. So with next generation sequencing, you start with the DNA of interest. That could be from an embryo, it could be from a miscarriage sample, it could be from an amniocentesis or a CVS, and you just fragment the DNA. You then take those little DNA fragments and you sequence them. After the sequencing is done, you'll now see the sequence for each fragment, and you'll be able to map it, realign it back to the genome. So you see a little piece of DNA, you see the sequence, and you say, oh, this little piece of DNA comes from chromosome A, I can map it back. And you do that for hundreds of thousands, typically millions of these little pieces of DNA, until eventually you then normalize to a reference, usually a normal male, to say, okay, we have the same relative abundance of DNA across all the chromosomes. So in the cartoon that I'm showing here, you have chromosome A in blue, chromosome B in red. You fragment, you sequence, you realign, and you look at the relative abundance, and they're equal. So you have the same number of copies of chromosome A as you do of chromosome B. In contrast, let's look at an example where you have trisomy of chromosome B. So 3 copies of chromosome B. You'd go through the same process. You take your DNA, you fragment it, you sequence it, you realign it to the genome. But this time, you have an increase, a 50% increase of reads that align to chromosome B. And that tells you you've got more reads aligned to chromosome B. That must mean you were starting off with more copies of chromosome B, and that allows you to use sequencing to detect aneuploidy. So if this is how aneuploidy works with next generation sequencing, why did we even bother looking at nanopore-based sequencing? And there's a few key advantages that nanopore-based sequencing has. The first is that it's much faster. It's actually over 15,000 times faster than next generation sequencing. So next generation sequencing sequences 0.1 nucleotides a second. Nanopore-based sequencing does 1,500 nucleotides per second. It's also much less expensive to start. Next generation sequencing setups begin at $250,000, but can often be over $1 million. Nanopore-based sequencing, you can set up for about $5,000. And the last major advantage is also the size. So I'm showing here a picture of a next generation sequencer, and below it is a picture of a nanopore-based sequencer. I also brought one with me, so I don't know if you can see in the camera, but in front of me, this is one of the nanopore-based sequencers that we use. So this sequences 15,000 times faster than the giant machines that cost $250,000. If you see this here, I'm not sure if it comes through on the camera, but the way it works is you open it. There's a little slide that goes in where you place your DNA. You close it. You plug it into your computer, it could be even a laptop, and you run it right on this device. The little chip that runs inside is disposable. It's reusable, but also disposable. So that's why we were interested in looking at nanopore-based sequencing. The problem with the existing methods, though, was that they used very, very long strands of DNA. That's what it was developed for. So what we realized was, for the purposes of what were important for reproductive medicine, we didn't need a long strand of DNA. We had already mapped out the human genome. We didn't need to keep redoing that. But what we did need was a lot of little reads so that we could map them all and do the counting exercise to figure out if we have the correct number of chromosomes or not. So over what's almost been a decade now, we've been developing this technology, and I'm citing 3 of the earlier papers on the method development below. But what we developed was this ability to do short read, transpore, rapid karyotyping, what we're calling STORC. So with traditional nanopore-based sequencing, you look at fragments of DNA that are about 40,000 nucleotides long or bigger. With STORC, we re-engineered the chemistry so that you can use half a kilobase or smaller. The regular library prep was very lengthy and complex to do. That's fine if you're in a high complexity research lab setting where you're trying to create the world's first assembly of, for example, a rice species. But if you're using clinical specimens where you need to do tens, hundreds, even thousands of these samples in a laboratory, you can't have a complex process that takes days to perform by a talented postdoc. You need something that's fast and simple, and that's what we developed. And the last criteria was a turnaround time. Traditional library prep and sequencing was over 18 hours to do. Our method takes less than 2 hours to perform, starting from the library prep through to sequencing. And this means that we can now sequence 1 million nucleate reads in 1 hour and test a sample in under 10 minutes. So we went from testing that used to take literally weeks to do to being able to get the results back in under 2 hours. In fact, when we were validating and developing the test, what was particularly exciting is we'd be able to test a sample and get the results back before the patient's clinical sample was even picked up by the courier to send for the traditional testing. This is a schematic showing the rate at which the sequencing happens. One of the other big things that we developed to help bring down the cost was the ability to multiplex, to be able to run many samples at the same time. So if a flow cell, for example, what I'm showing you here, costs $500 to use, to test one sample, that's expensive. But if you could put 10 or 20 samples and run them at the same time, suddenly your costs are going to be much, much lower. This is showing how quickly we're able to generate the reads. So to be able to diagnose aneuploidy, we need about 80,000 reads. And this is sort of showing that if you use one sample, it's really about 7 minutes, we get the results. But if you run 10 samples plus the necessary controls, you can easily do it in about an hour to an hour and a half. The next big thing that we were working on was the method development, so that doing the actual library prep, getting your sample ready to do the sequencing could become quicker. So I'm showing here the relative timeline. Something like GBank karyotyping, you're looking at more than 270 hours. But even things like microarray and next generation sequencing, you're still well beyond the range that can give you same day or even within a few hour results. Stork, which I'm showing you down here, allows us to have that very rapid library preparation, sequencing, and data analysis pipeline. So the question was, the technology seems to make sense. There's obvious advantages to wanting to use it. And we showed that we were able to adapt the technology to do short read sequencing. But the big question is, does it actually work well? So for that, we did this clinical validation study, what Dr. Ash mentioned at the beginning. This study involved taking 260 samples across all the different types of reproductive tissues: amniocentesis, miscarriage samples, CVS samples, biopsies from an embryo, doing our library preparation, sequencing, analysis, and then looking at the results. When you look at the results here, this is actually how the data looks. So this is an example where you see an overabundance of a double, actually, of the X chromosome, because we always normalize to a normal male. So a normal female will have twice as much of the X chromosome as a normal male will. Then you see a similar amount of relative DNA across chromosome 1, 2, 3, 4, 5, 6, 7, all the way up until chromosome 13. You see an increase in chromosome 13 and a normal amount of the rest. So this is an example of a case that was a 47XX with an extra copy of chromosome 13. So for our validation study, we took these 218 blinded samples that were actual clinical samples that had been used and tested in the standard clinical workflow. We took the excess, the remnant DNA, and we subjected it to Stork sequencing, and then looked at the results, and then unblinded our results. So everything that we did, we were blinded on it. And this is how the results looked. For CVS, Amni, POC, and PGTA, we had a sensitivity of, well, for the first three of 100% and specificity of 100%. For PGTA, which there's always a little bit more of a question, it was at 95% sensitivity, 100% specificity, and you see really outstanding positive and negative predictive values. I do want to draw your attention to one point, though. If you look at the testing of the miscarriage sample, there were actually 10 discordant samples that we saw. 10 samples where the result we got with Stork was different from the results that the clinical laboratory had on the miscarriage sample. What we did in those cases is we told the clinical laboratory, right, once we unblinded them, like, okay, here there's a discrepancy. And the clinical laboratory went back and retested the samples. In every one of those cases, the results that the clinical laboratory had initially given were actually the wrong results. And the Stork results were validated by the retesting of the samples by the clinical lab. I think that was an important lesson, because anytime you compare it to something else, if the something else isn't perfect, if there's a discrepancy, you have to find out where the problem actually rests. So here's an example of where there was a misidentification, where a discrepancy, where we had one result and the clinical lab had a different result. So we ran our sequencing, and as you see here, a doubling of the X chromosome, so you know this was a female sample, and an increase in chromosome number 9. This was the result that the clinical lab gave, where they said trisomy 8. But then they reanalyzed the sample and they ran it with a microarray and confirmed that that sample was actually an extra copy of chromosome 9. Now, all of you, I'm sure, are looking at that and you're like, okay, well, that's interesting, but that's not so clinically significant. If you tell a patient she had an extra copy of chromosome 8 or 9, that's not such a big difference. And that's true. But here are cases where it was a big difference. So here's an example where the results that we got with the Stork nanopore-based sequencing showed that you had a normal number of X and Y chromosomes. So this was a male sample with an increase in chromosome number 7. So this was a trisomy 7 sample, a finding that is incompatible with life. But the results that the patient had been given was that it was a normal sample, a normal male sample. So they went back and used a different probe for chromosome 7. And sure enough, when the lab retested their sample, it turned out that there was a mutation in the patient's chromosome 7 that prevented their probe from binding to it. But in fact, it was trisomy for chromosome 7. And that has very real world implications for the patient. If you find out that the loss was genetically abnormal, you have your answer, you have your explanation for why the miscarriage occurred. That's the cause for the majority of miscarriages. But if the result comes back as normal, it means there's something else going on, and that triggers a whole investigation with blood tests and imaging and oftentimes medications and treatments that aren't always benign. Here's another example of a discrepancy. In this case, Stork showed that the result was normal, a normal male. And you see across all the chromosomes, a relatively identical number of chromosomes. But then when we look at the report that the lab gave, they told the patient that they actually had an abnormal sample, an extra copy of chromosome 15. They repeated that sample and looked at it with G-band karyotyping and microarray, and again confirmed that in fact there was an error in the probe. The probe had misaligned to another chromosome, and there were in fact only 2 copies of chromosome 15. This was a genetically normal pregnancy, and identifying that early is important because it allows you now to figure out what was the real cause of the loss and hopefully correct that before the woman will have another pregnancy loss. And how does this method of Stork compare with other methods that are out there? So here I'm showing the different methods that we talked about: G-band karyotyping, FISH, microarray, next generation sequencing. I'm looking at the setup costs, the reagent costs, the turnaround time, whether sample transport is required or not, and the location of testing, as well as the number of chromosomes tested and whether you need living cells. And you can see the big advantage that Stork has is it's much less expensive to set up. It costs a lot less per sample. You get the results back in days. Instead of taking days or weeks, you get the results back in hours. You can do the testing on site. You don't have to ship it to a centralized lab. You test all the chromosomes, not just a subset, and you don't need living cells, which is important if you're testing a miscarriage sample that might be older or tissue that's been paraffin embedded or fixed. But like every technology, there are limitations to Stork. 3 of the main ones are that, number 1, the resolution is lower than SNP microarray, so its ability to detect duplications or deletions is going to be reduced. The other thing is it cannot detect inversions or balanced translocations. Microarray, next generation sequencing, they can't detect it either. Only G-band karyotyping really can do that. And the ability of this platform to recognize triploidy is something we're still continuing to work on, where you have 3 copies of every chromosome. That's a similar limitation to what you're seeing with traditional next generation sequencing and array CGH. So what are our conclusions from this? The 3 key ones are that, number 1, Stork does have this potential to provide rapid, low-cost point-of-care testing for aneuploidy across all reproductive sample types. It can expand access and reduce barriers to care. And like with every other technology, there are limitations to it. I'm also happy to be able to share with all of you the news that New York State, which has the highest regulatory burden in the US, has now approved stork for clinical testing of miscarriage samples. So that's something that just happened. And it's very exciting because it means we can now actually offer it to patients. It's past the most stringent regulatory hurdles that we have in the US. So I wouldn't be complete without thanking the team that helped do this work. Vivian Way, a postdoc and now junior faculty in our department, and Hemant, another faculty in our department, Bren Levy, who worked a lot on the clinical side of the genetic testing, Ron Wapner, and Alex, who is a member of the IVF Center. And of course, I want to thank the NIH, who so generously funded this work. And just want to end with a picture of the Fertility Center team that we have up in New York, and to extend a welcome to all of you. You're all cordially invited to come to New York, to Manhattan. We're located in Midtown in Columbus Circle by Central Park. There's great ice cream next door. And as long as you wait for the Canadian fires to stop and so we don't have the same air issues, I think you'll have a wonderful visit. Thank you very much.